Welcome to this video on lipids of biochemical importance, part of Module BC 2103. In this video, we will explore the structure, properties, and biological significance of various lipid types. Here is an overview of the topics we will cover: the general importance of lipids, followed by a breakdown of fatty acids, triacylglycerols, phospholipids, glycolipids, and steroids. Lipids are a diverse group of molecules that are largely insoluble in water, but soluble in nonpolar solvents. They are essential for energy storage, cellular structure, and cell-to-cell signaling. Lipids can be classified based on their structure. Many, such as waxes and triacylglycerols, are esters that can be hydrolyzed. Steroids, however, have a unique ring structure and cannot be hydrolyzed. This diagram illustrates the classification of lipids, showing the relationship between fatty acids, steroids, and their various derivatives like waxes and glycerophospholipids. Fatty acids are long carbon chains with a carboxylic acid group at one end. They are typically unbranched and often contain an even number of carbon atoms, as seen in this example of lauric acid. Fatty acids are categorized as saturated, containing only single bonds, or unsaturated, containing one or more double bonds. These double bonds create cis or trans isomers, which significantly affect the molecule's shape. This slide displays structures of polyunsaturated fatty acids like linoleic and arachidonic acid. These are essential fatty acids that must be obtained through our diet. The physical properties of fatty acids, such as their melting points, are determined by their structure. Saturated fatty acids stack neatly, leading to higher melting points, while unsaturated ones contain kinks that lower these points. This visual comparison demonstrates why saturated fatty acids, like stearic acid, are solid at room temperature, while unsaturated ones, like oleic acid, are typically liquid. Waxes are another type of lipid, formed by the esterification of long-chain fatty acids and alcohols. They serve protective functions in plants and animals by providing waterproof coatings. Here we see common examples of waxes, such as beeswax, carnauba wax, and jojoba wax, along with their various commercial and industrial applications. Triacylglycerols, or triglycerides, are the primary way our bodies store fatty acids. They consist of a glycerol backbone esterified to 3 fatty acid chains. When glycerol reacts with 3 fatty acids, it forms a triacylglycerol. Most natural fats contain a mixture of different fatty acids, as shown in this structure. Finally, triacylglycerols are crucial for energy storage, especially in animals that hibernate. They allow these animals to survive for months by metabolizing stored fat when food sources are unavailable. Moving to chemical properties, hydrogenation is a process where hydrogen gas is added to the carbon-carbon double bonds of an oil in the presence of a nickel catalyst, converting them into single bonds. In commercial production, partial hydrogenation is used to transform liquid vegetable oils into semi-solid fats like margarine and shortening. These products lack cholesterol, unlike butter or lard. Hydrogenation can lead to the formation of trans fatty acids, which have a more stable structure. Research suggests these trans fats may raise LDL cholesterol and lower HDL cholesterol levels. Trans fats are naturally found in some animal products and are also present in many processed foods. Health organizations recommend reducing their intake by choosing liquid oils and limiting saturated fats. Hydrolysis of triacylglycerols involves splitting ester bonds with water in the presence of acids or digestive lipases, producing glycerol and 3 fatty acids. Saponification is the reaction of a fat with a strong base like sodium hydroxide to form soap. Different bases, such as potassium hydroxide, yield softer or liquid soaps. Phospholipids are structural lipids similar to triacylglycerols. They include glycerophospholipids, where a phosphate group is attached, and sphingomyelins, which use a sphingosine backbone instead of glycerol. Amino alcohols like choline and ethanolamine are often found in glycerophospholipids. Lecithins and cephalins are examples that are particularly abundant in nerve and brain tissue. Glycerophospholipids are amphipathic, meaning they have a polar head and non-polar tails. This allows them to effectively interact with both water and lipid environments. Glycolipids contain carbohydrate chains attached to a lipid backbone. They are critical for cell-to-cell recognition and are found primarily on the outer surface of cell membranes. Chemically, glycolipids are characterized by a sphingosine or glycerol backbone, a non-polar lipid tail, and a polar carbohydrate head group connected by a glycosidic bond. Steroids are defined by their unique nucleus of 3 cyclohexane rings and 1 cyclopentane ring. Cholesterol is a key example, vital for membrane structure and precursor for other steroids. Cholesterol plays many roles, from forming the myelin sheath to acting as a building block for bile salts and steroid hormones. While essential, excess levels are a health concern. Elevated cholesterol levels can lead to the formation of plaque in arteries, known as atherosclerosis, which may restrict blood flow and increase the risk of heart attacks. Thank you for watching this overview of important lipids and their biological functions. Understanding these molecules is key to grasping core biochemical processes.